Hello, everybody. Here we are on week 4 of our anatomy and physiology course. I hope you're all doing well. We have two things to accomplish this week, September 20th, and that is one is you have to take quiz 1 this week. You click on the quiz 1 on the panel on the left of Blackboard, and it will appear, and you just click on it to start, and then submit your answers. Okay, it has to be done this week. Sometime before Friday, and you only get a chance to take it once, and you have an hour and a half to do it. You can use whatever materials you want to take the quiz with. It's multiple choice, 30 questions. And as soon as you're done, you just click on submit, and you're all finished. The next thing is the cells. Now, the cells is our next topic. And those are the living things that make up the human body, a couple of trillion of them. And they are alive. They're the things that actually are alive inside of you and do all the processes of life. The questions on cells, survival guide questions, click on them, and they will be also due this week. All right, they'll be another part of your final exam. So as we accumulate these final exam grades, you'll have 20 points by the end of the course, and that'll count as your final exam. So for this week, again, you have two things to do. One is to take quiz 1, and 2 is to hand in the cell questions the way you've been doing it, using the assignment section on Blackboard. Some of you have been having some trouble with that assignment hand-in thing. It might be because of the computer you're using or the type of Chrome you have, whatever operating system. I had to get a new computer because mine didn't work either until I got one that was more up to date. So you might have to update your operating system or your web browser or whatever to get it to work better. Some of you handed in things on email and that worked, but I'm just a little concerned that you won't be able to take the quiz. So if anything's a problem, just give me an email and we'll talk about it. Okay, now as far as cells go, we have a short recording we're going to make on the parts of a cell and what they all do. So I'm going to start sharing my screen with you now and showing you the PowerPoint that you would have seen in class. It says the cell is the unit of structure and function of all life forms on the earth. What that means is that every life form on the planet that we know of is either made of one cell or many cells working together as a team. What happens in terms of life is what cells do. The question is, are you alive? Obviously you are, but you can actually argue that no, I'm not alive, my cells are. Each cell in your body has a lifespan. It is born, it lives for a while, then it dies and gets replaced. There's only a few organs in your body that have cells in them that are the same cells you were born with, the brain be one of them. But most of the other cells in your body are constantly dying and being replaced. They have a life, they live inside of you, they get born and they die. The cells then, where do they come from? And so far, we can't make a cell in a test tube yet. But so far, the only thing that can make a cell is a cell. And what they do is they do a thing called binary fission, or they do a thing called mitosis, where one cell can become two. Binary fission is not our problem. That's because we're not a microbiology class. But binary fission is basically what bacteria do to reproduce. They simply just replicate their DNA. Remember we said that one of the functions of DNA is that it can make copies of itself. Replicate the DNA, separate the replicates, and then pinch in between. And you end up with two cells. A very simple process. It takes only about a half an hour. And one bacterial cell can become many, many millions very quickly. That's not what we do. We do a thing called mitosis. Mitosis is our way of replicating our cells. And what happens is that the nucleus of the cell, which we'll see in a few minutes when we talk about the cell parts, can replicate. The DNA can copy itself, separate the copies, and then the nucleus will pinch down to become two nuclei, and then the cell will pinch in between the nuclei and end up with two cells, each with one nucleus inside. This is not a biology class, so we don't have to really worry about all the steps of mitosis. But all I'd like you to know is that our cells reproduce by that process. And what they do, again, is they just replicate their DNA, replicate the nucleus of the cell, and then actually pinch the cell up to create two cells with each with a nucleus inside. You're doing that right now in many, many parts of your body. It's done to create new skin cells. It's going to grow your hair and fingernails and replace cells that die in your body, and also heal wounds and things like that. You're doing it all the time. The next slide shows you my diagram of a typical animal cell. And you can see it looks kind of, it's basically a structure that contains smaller parts, and each one of the smaller parts has a different job. And so our lecture today is basically going to depend on what each one of those cell parts actually do for the cell. And when they all work together as a team, the cell is actually considered to be a living thing and can do all the processes of life. So what we're going to do is we'll go through the different types of, they call them organelles or small organs in the cell. And we're just going to discuss what each one of them does. So in the next slide, I actually showed you the same picture, but with all the parts labeled and described. This will be like 90% of what you need to know for this lecture. Basically is what each part of the cell does and what they are made of, what they look like. Number one is the mitochondrion. A mitochondrion is those little yellow things in there that look like little smiles. And what they are, they have two membranes, an outer membrane and an inner membrane that folds all up that look like little teeth. And inside the mitochondria are lots and lots of enzymes and biochemical reactions that allow you to break down your food, especially things like glucose and amino acids from proteins. You break down your food and release the energy from the food. The food is broken down all the way to just molecules of carbon dioxide, CO2, and water. And the carbon dioxide and water, the water becomes part of the water of your body, and the carbon dioxide you breathe out through your nose and mouth every time you exhale. The mitochondria are the source of all that carbon dioxide that you breathe out. Also, the mitochondria is why you need to breathe oxygen. The oxygen is sent to the mitochondria to allow the mitochondria to literally burn or combust the food that you have eaten. So when you eat, you break down the food in your digestive system to very, very, very small particles. It's particles small enough to get into a cell. Your bloodstream will dissolve in your bloodstream. Bloodstream will take the food particles, the amino acids, the glucose, even some of the fatty things, and bring them to the cells. And within the cells, most of the food goes into the mitochondria. And what the mitochondria will do is react the food with the oxygen that you're breathing and literally burn it in a process that we call cellular respiration. Cellular respiration is a slow burning, using enzymes, many, many steps, to actually convert the food molecules, the glucose, amino acids, etc., into carbon dioxide and water. And just like when anything else burns with oxygen, it releases energy. And the energy is used to do two things. One is to keep you warm. Your body temperature is 98, but you very rarely live in an environment that's 98 degrees. So, how do you keep your body temperature at 98 when you're in a room that's 85? You do that by using the heat generated by the mitochondria as it's burning up the food you've eaten. The rest of the energy, about 30% of it in your food, is not converted directly to heat. Instead, it's used to make ATP. And what it does, it uses the energy to add a phosphate onto an ADP. That's adenosine with two phosphates, and then put that third phosphate on there and make TP, so that you can use those ATPs, or adenosine triphosphates, as an energy source by breaking the last phosphate off and using the energy in that broken bond to basically power all the things in your body that you need energy for. Things like your heartbeat, breathing rate, using your pulse, all of those things that you're constantly requiring energy for. Even thinking a thought requires ATP. Blinking your eyes costs you so many ATPs. And so you got to constantly be making these things. And that's why you eat the food you eat and digest it to send it to the mitochondria, and the oxygen that you breathe also to be sent to the mitochondria, so that the food and the oxygen can react in cellular respiration, turn the food into carbon dioxide and water, and the energy released is either released as heat to keep you warm or to make those ATPs that you literally live on. Now, the mitochondria really are actually bacteria. That's why there's two membranes. There's an outer membrane, and then the inner membrane is really a bacterial membrane that the little bacterial cell is literally living inside the outer membrane. And so you have each mitochondrion in your body, and you have hundreds of your cells. There's only 6 in my diagram there, but you have hundreds in your cells. They're little bacteria that are actually living inside of you, and they're the things that you actually use to burn up your food and release the ATPs and the heat. So that's the mitochondria, and what they do is cellular respiration, as it says there, which is the process of literally reacting your food with oxygen to create carbon dioxide and water as a waste, and also release the energy as heat and ATP. The second one is the nucleus. The nucleus is that middle part cell there that looks like it's kind of bluish in the diagram. And what it is, it's kind of, if you look at the cell membrane, you can see this outer cell membrane is kind of folding in. And as it's folding in, it's creating these channels. And the channels are kind of pushing all that material in the nucleus together in one spot. The material inside the nucleus is basically made of DNA and protein. So what you're doing there is putting all the DNA and the RNA and all the enzymes that it works with together all in one place. And that's called compartmentalization. It creates a lot of efficiency. It's like having a classroom. Remember classrooms? It's having a classroom where your students are all compartmentalized in one room, and you can teach 25 or 30 of them at a time very efficiently, as opposed to if they were spread out all throughout the building. The nucleus then contains the DNA in the form of something called chromosomes, and each chromosome is made of DNA and protein, and there's hundreds and hundreds of genes on each chromosome. Humans have a set of 23 chromosomes that makes a complete set of them, and each chromosome has hundreds of genes on it, or many genes on it, depending on the size of the chromosome. Each gene has enough information on the DNA to produce one protein. And so a gene enough information on the DNA to teach the cell how to put amino acids together in the proper number and order to create the protein that the cell needs. And you have thousands of genes on 23 chromosomes. Now, in human beings, of course, we have two parents, so we have two sets of 23. We have 23 from our mom, and we have 23 from our dad. And every nucleus in your body contains 2 of each chromosome, one from your mom and one from your dad. And that's good, because what happens there is that if you have a problem on a chromosome, a gene that doesn't work, or it's mutated, or something's wrong with it, the one on the other parent's set of genes or chromosomes will back that up. And that's why we could have so many genes. And of course, when you have lots and lots of genes working, it's more likely you're going to find something that doesn't work. Each one of the mistakes on one set of chromosomes are backed up by the other one. And that's why the two parents should not be too closely related, so that the two sets of DNA wouldn't be that similar. So there would be a mistake in the same place on both of them. So that's why incest is not a good idea. But the thing is, though, that's what's inside the nucleus. It's all the DNA and the RNA and genes and enzymes and things that need to work together for the genetics of the cell to work. And if you put them all together in one place, compartmentalize them in the cell, it's much more efficient that they can work together. And so that's the nucleus. Number 3 is the nucleolus. The nucleolus is that little pink thing inside the nucleus, and you see there's 2 or 3 of them there. What they are little sort of particles made of RNA and protein. And what they do is they produce little tiny pieces of RNA and protein that actually leave the nucleus and populate the rest of the cell. Those little pieces of RNA and protein are known as ribosomes. And we'll talk about those as we get down the list here. It's number 10 on the list if you look at it. A ribosome is the very, very small little chunk of RNA and protein that actually attaches to the messenger RNA that comes from the nucleus that has the information on how to make a protein from the DNA, and reads the RNA and literally puts the amino acids together to create the protein. So the ribosomes, which are made by the nucleolus, are actually the locations in the cell where the proteins are actually assembled and put together. And that's the nucleolus, and it's inside, they're inside the nucleus making those ribosomes. The next one is called the endoplasmic reticulum. Now, those are those folds of the cell membrane that look like little channels or canals inside the cell. The cell membrane will fold up to create these channels in the cell, and they're all branchy. So endoplasmic just means in the cell, and reticulum or reticulated anything means it's very branchy. So a branched network of channels in the cell is the endoplasmic reticulum. Now, what it's for is for transport. It's kind of like the Route 95 of the cell. It's kind of like a highway where things are a little more liquidy in there. They can move around a little better inside the cell to get from one part of the cell to the other. And also things on the membrane of the endoplasmic reticulum, you could have enzymes. So as something goes by in the ER to go from one spot to another, as it passes an enzyme that's kind of attached to the membranes, the enzyme can modify it. So as things move along in the ER, they can be changed chemically using the enzymes that are embedded in the endoplasmic reticulum membrane. So we say not only as the endoplasmic reticulum for transport of things around and throughout the cell, but it's also for modifying them as they go along. So it'll be like almost like a Route 95 can fix your flat tire as you were driving on it. The endoplasmic reticulum, as you see, also forms the nucleus as it squeezes all those nuclear materials together in one spot. Now, you could also use the endoplasmic reticulum for storage. You notice that some of the branches of the endoplasmic reticulum or ER sort of end in a dead end. So you can put materials in there and just let them sit there, just like you park your car in the driveway, and wait to use them later. You'll see when we talk about muscles that's very, very important in muscles. There's something stored in the endoplasmic reticulum dead ends in the muscle cells that actually help the muscles contract, and we'll talk about those in a few weeks. But that's storage. So the three jobs that the endoplasmic reticulum have is to be a transport system. It can modify things as the things go through it with enzymes that are embedded in the membrane, and also for storage. You can store things in those dead ends. Now, there's two types of ER. There's smooth ER, which looks like just doesn't have any hard spots on it. And then there's rough ER that does look like it has spots on it. And if you look at the diagram, the smooth ER is the ones that don't have those little dots. If you look at the left side, you'll see one that has, says number 5, it has little dots on it. Those little dots are ribosomes, the same ribosomes I talked about a little while ago where proteins are made. So what's probably transported, modified, and stored in rough ER, they call it rough because it looks bumpy with an endoplasm, with a electron microscope, because it has ribosomes on it. What's stored in the rough ER would be mostly proteins and modified and transported. In the smooth ER, it would probably be things that are not proteins, things like lipids and carbohydrates and other steroids and are not proteins. Lipids, triglycerides, those kinds of materials can be moved around in the smooth ER and modified. So that's the endoplasmic reticulum. And in a real cell, it's probably going to be much more extensive than it looks like in this diagram. Then you get to the next one, number 6. Number 6 is the Golgi, and the Golgi is actually named after someone named Golgi, and that's why we always use a capital G for that. But the Golgi are those little blue things that look like stacks of pancakes in there. The Golgi are basically pieces of endoplasmic reticulum that have broken off and stacked up. They're loaded with enzymes in them, and enzymes that are attached to their membranes. And what they do is they manufacture all sorts of materials that the cell needs. Not only do they manufacture them, but they can store them in these little sort of what they call vesicles. And if you look at the Golgi, you'll see little circles kind of surrounding them, little gray circles. Those are called Golgi vesicles. And whatever the Golgi is making, they would pop off the Golgi as those vesicles, migrate to the cell, and end up at the cell membrane. And when they get to the cell membrane, they can attach to the cell membrane and open up and let whatever the Golgi was making out of the cell. That is a way for materials to get out of the cell without literally having to go through the cell membrane. So what happens is that the vesicles actually become part of the cell membrane. They open up and then let that material out. That process is called secretion. So if you have a gland that has to make things like and hormones or sweat or whatever it might be, or tears, the Golgi is secreting that to the outside of those cells, and that material is leaving the cells. And if it's a gland like the pancreas, it would be putting the insulin into the blood, and if it's like a tear gland, it would be making the tears out get to the outside of the gland so they can wash your eye. And so that is what the Golgi do. They secrete. They make materials, and the materials are stored in those little vesicles. The vesicles move toward the cell membrane, open up, and then the material is secreted to the outside of the cell. So we say the Golgi is basically for manufacture, transport, and secretion. It'll make something, transport it in those little vesicles, and then let it out of the cell. And it can be many glands are using Golgi right now in your body to secrete the materials that the cells are making, either to the outside of your body, or even into your stomach, or into your blood. Okay, now, next one is the lysosome. The lysosomes are also modified ER. They break off the end of the reticulum, and they have inside of them about 30 or so different digestive enzymes. And what the lysosomes do is they destroy things. Lyse means to literally destroy something. And what they'll do is they will, if the cell is, let's say, attacked by a virus or cancer or something like that, the lysosome can pop, and those digestive enzymes for digesting proteins, carbohydrates, lipids can leave the lysosome and literally digest away the cell and kill it. So lysosomes are basically almost like little hand grenades inside the cell that actually have the power to destroy the cell if the cell becomes a problem. Also, if the cell is like a white blood cell and it's eating things like bacteria in your body, the lysosomes can digest and kill the bacteria within the white blood cell as well. If one of the mitochondria dies, because remember, mitochondria are bacteria too, they can be consumed by the lysosome. In the sense, it's like the little stomach of the cell in a way. So they contain digestive enzymes. So if you look at the lysosome word, it says digestion next to it. Not found in plants, only found in animal cells. Then we have the vacuole, number 8. The vacuole is really membrane-bounded sac, also broken off the endoplasmic reticulum, that is used to store things. You can store water in there, you can store waste, you can store food. It's like a closet, and you put things in there that the cell needs later on. So the vacuoles in animal cells, anyway, it's used mostly for storage. In plant cells, they're much bigger, and they're used to store water and create pressure within the cell, so plants can stand up without any wood, if they're not woody plants. They can create a lot of water pressure within the cell. But in animal cells, they're small, and they're basically used for storage. Next one are the centrioles. They look like those little look like a little cross in the middle of the cell there, number 9. The centrioles are basically also degenerated bacteria. What they do is they live inside the cell, and they can manufacture something that certain types of bacteria do, but we're not. And that is something called microtubules. A microtubule is a little protein sort of tube. It's very small and hollow. It's probably the smallest hollow thing in the world, and they're perfectly straight and almost like little things, but very small and long and thin. And what they do is they populate the cell. Now remember, everything outside the nucleus is called the cytoplasm of the cell. And they're within the cytoplasm, these microtubules that the centrioles make. And what they create, they sort of interlock within the cytoplasm and create sort of a lattice, almost like a skeleton. And they give the cell some structure so the cell can have some support. It's almost like the cell's bones in a way. So we actually call what the microtubules become, we call them the cell skeleton or the cytoskeleton. They support the cell. And in my diagram here, they're hard to see, but they're in there. And there's a lot more of them in a real cell. If I put just all the microtubules all stuck together in there, you wouldn't be able to probably see much of anything else. But they are a big lattice work within the cell that creates a skeleton for the support. Also, some of the organelles can actually walk along those microtubules and move around inside the cell as well. Use them as a little track. And also, when the chromosomes separate, when DNA replicates and separates the genes, when the cell is going to replicate, the chromosomes move down a track made out of microtubules to help the cell separate the chromosomes so that you can create two nuclei and eventually two cells. So those are the microtubules. They're made by the centrioles. Centrioles make the microtubules. Now, if you look at the number, I think it's number 9, again, over on the right side, where that little thing is sticking out of the cell, that is number 12 there. That's also a centriole. It's a little different color pink, and that's not an issue. It's just the way the computer made it. But the centriole is making microtubules, but this time the microtubules, instead of just populating the cell and creating a skeleton, instead they're poking at the cell membrane. They're growing out of the centriole, and notice the cell membrane itself is actually wrapped around that sort of green rod in there. The green rod is made of microtubules. And it turns out that microtubules, if they're right next to each other, there's many of them in there, little proteins on them called dynein proteins, which actually can walk like little feet using ATP. And so the microtubules kind of walk over each other inside that number 12. And that causes that little sort of part of the cell that's extending out, that extension to wiggle. And we call that cilia, C-I-L-I-A. One of them would be a cilium. And if this was an actual living cell, you'd actually see that going back and forth. It would be wiggling. And there would be many of them on the cell, not just one. For example, inside of your trachea, the part of the body where you breathe oxygen down into your lungs, it's lined with cells that have cilia. It almost looks like grass when you look at it under a microscope. There's so many cilia. And what they do is they beat upwards. And so you have mucus being formed in your trachea all the time to catch any bacteria or dust particles or whatever it is that you're breathing in so they don't reach the lungs. And then when the materials that you're breathing in, those particles that could hurt you, they get stuck in the mucus. And then the cilia beat in the mucus and push them up toward your throat. And then you swallow your saliva about, once every minute or so. And when you're swallowing that, you're also swallowing what's coming up from your throat with the cilia, pushing up the dirty mucus and down into your stomach. And your stomach kills all the bacteria and things that are trying to hurt you. That's one of your first lines of defense against disease, especially pneumonia in your lungs. So cilia are little extensions of the cell membrane. The cell membranes are wrapped around the cilia. And the inside of that is microtubules made by the centrioles that actually wiggle within the cilia. If the cilia is very, very, very long, longer than the cell by many times, then it's called an undulopodium. And it looks like a big tail on the cell. The old name for that used to be flagella. When an undulopodia wiggles, it causes the cell to swim. And the only cells like that in the human body are sperm actually made by the male. Nothing like that in the female. And so if this was a sperm cell, that number 12 there would be very, very, very long. It would be called an undulopodium because it undulates, goes back, it just wiggles. And it causes the sperm to swim toward the egg when people have sex. And so that's the undulopodium. The last thing on the list here is the cell membrane. The cell membrane is wrapped around the cell. And learned from the last lecture, it's made of 2 layers of phospholipid molecules with some cholesterol embedded in between them. The phospholipid molecules are not connected. They're held together by the positive ends of water molecules on either side of the cell, by little magnetic water molecules. The cell membrane phospholipids can actually change places with each other in the membrane, so it actually flows around the cell like it was a liquid, very much like a soap bubble. You know that a soap bubble looks like it's a solid thing, but if you look at it carefully while it's floating around in the air, it looks like things are sort of moving around inside of it, inside of the soap bubble's membrane, because it really is a liquid. And the same thing is true here for the cell membrane. The cell membrane is just 2 molecules thick. It's a fluid membrane. It actually flows. That's why the endoplasmic reticulum can fold in like that. It's not a solid thing. Things are changing all the time in any one of your cells. And what does the cell membrane do? Well, obviously, it's the boundary between the inside and the outside of the cell. Certain things like very small molecules that don't have a lot of charge on them, like oxygen and carbon dioxide, they can go right through the membrane like it wasn't even there. And so the cell can get that stuff just by having it fuse in and diffuse out if it's carbon dioxide. Bigger molecules, well, in the membrane, there's proteins embedded in there, and we talked about that when we talked about proteins last time around. And the proteins can actually pump the things through the membrane, into the cell, or out of the cell, depending on what the cell needs to do. There's various protein pumps for different things. There might be protein pumps for calcium ions, protein pumps for amino acids, or whatever it might be. And the cells can use those, powered by ATP, to pump things into the cell or out of the cell, depending on what they are. If they don't work right, you can get lots of genetic diseases. For example, cystic fibrosis is caused by a defective pump that doesn't pump the calcium ions into or out of your lungs properly. And so you get all kinds of trouble with breathing, with other organs too. So anyway, that's the cell membrane's boundary. It can control what goes in and out of the cell. It also has on the outside of it proteins that actually act as identification proteins. In other words, outside the cell on the membrane, there are protein molecules that can tell your immune system that those cells are actually you, so that your immune system doesn't attack them and destroy them like they would a foreign cell. So in the cell, your cells have, your cells have a uniform around them that tells your body that you're the good guys. And that uniform are protein molecules called identification proteins that actually teach the immune system to leave that cell alone and to kill other cells that don't have the proper identification proteins. Your blood typing are identification proteins. The cells then have all of these parts, and they all work together as a team. And of course, that creates the cell. And if you remember from the first lecture, we said you're made out of chemicals, and the chemicals get put together to make bigger chemicals like proteins and carbohydrates and lipids and DNA and ATP and RNA. And they can get put together to make these organelles. And then the organelles work together to make the cell. And now next week, we're going to talk about how cells work together to create tissues or groups of cells that have jobs to do in your body. But right now, we're still working on just one cell. The next slide is just a slide from a book, and you can actually see some of the cell parts in 3D. And then finally, you have these 2 slides. One of the questions on your homework this week is going to be to list the parts of the eukaryotic cell. Eukaryotic cell means a cell that has a nucleus, and what each part does. And these 2 slides, this one and the one after it, that one, are going to be the answers to those questions for you. Okay. Eukaryotic cells are cells that contain a true nucleus. That's our cells, human cells, animal cells, plant cells. Bacteria don't have a nucleus, so they call those prokaryotic cells, a cell without a nucleus. Their DNA is just floating around inside the cell with the chemicals that they need to work with. But bacterial cells are so much smaller that they can get away with that. Now, This is a cell membrane. We're almost done now. This is a cell membrane. You can see the two layers of phospholipids. They look like those two layers of lollipops in there. You'll notice that the little gray circles are the glycerol phosphate portion of the phospholipids. Those are negatively charged, and that's why there's all those little negative signs on them on both sides. Now, if there's water around, the positive end of the water molecule, the Mickey Mouse ears, will actually be attracted to those negative phospholipids and hold them in place. The phospholipid, the whole cell membrane of your body is held together by the polarity of water. That plus side of the water molecule, little magnet, is actually holding everything together because the phospholipids have a minus side. That's the two layers of phospholipids they create the basis of the membrane, and they go all the way around the cell. And then you notice that embedded in the cell, there's different kinds of proteins. One's called a facilitated diffusion protein. That means that it basically has a hole in it, so various substances of the right shape can actually go through that hole and get into or out of the cell, depending on what kind of material they are. Some facilitated diffusion proteins cause things to leave the cell, and some cause them to enter. It depends on what it is. If you notice the red one, that is a receptor protein. That's a protein waiting for a chemical to attach to it, and that causes something to happen within the cell. It's almost like if you push a doorbell, you push it on the outside of the building, and your finger is attaching to the doorbell, but that's causing something to happen within the building. The doorbell rings. And so the receptor protein, you're going to see that's a big deal in nerves and muscles, where you're going to find that molecules have to attach to receptors on the cell membranes of muscles and nerves in order to create an action within the a muscle or nerve cell, to make you able to move or to even think a thought. So receptor proteins are just there to wait for a protein to attach to them or a chemical to attach to them in order to cause an effect within the cell. The active transport carriers, those are the ones that use ATP to pump things into the cell or out of the cell, depending on what the cell needs. And then finally, those little green things that look like spaceships sitting on the outside the cell there, those are the identification proteins that actually tell your immune system what kind of cell that is. And so you can avoid destroying your own body with your immune system. And if you look at the other side of the cell membrane, you see there's enzymes in there. And almost every cell membrane in your body, even including the endoplasmic reticulum membranes, which are also made of the same material here, they have enzymes attached to them that can cause chemical reactions to happen in the cell as they need to happen. And if they're attached to the cell membrane, and if you have a series of chemical reactions that have to happen one after another, then enzyme 1 can do the first reaction, and then enzyme 2 can do the second reaction, enzyme 2 can do the third reaction, and it's very, very efficient if those enzymes are attached in sequence on the cell membrane itself, on the inside of the cell. So that's basically the cell membrane. You can see it's a very complex thing. It has lots of jobs to do. And you can see also there's microtubules there holding everything together as part of the cell skeleton. And then finally, we have diffusion and osmosis. Diffusion is nothing more than spreading things out. For example, if you have something that's very smelly, it will eventually fill the room with its odor. If you're far away from it, you can't smell it, but a little bit later on you can. Just molecules, they always move from where there's more to where there's less of them. If you put a drop of food coloring in a glass of water, at first it's just in one spot, and you see that nice red spot or whatever color it is, and then eventually, if you leave it alone, it'll spread out through the entire glass and create an even color throughout the whole glass of water. That's diffusion. It doesn't cost the cell any energy. It's just it uses the energy of the molecules themselves that are moving because of their heat energy that they have inside of them. If something can get through the membrane, like oxygen, if there's more on one side of the cell and less on the other side, it'll just diffuse right into, most of the time, into the cell. Carbon dioxide is made by the cell because the mitochondria make that, and it will accumulate in the cell and simply diffuse out as it spreads out through the membrane. They can get those, remember, those small molecules that don't have a lot of electrical charge can actually move right through those phospholipids and get out of the, into or out of the cell through the membrane. Osmosis is just a diffusion of water across the membrane. If you have more water on the outside of the cell and less on the inside, water will spread into the cell and cause it to swell. If you have less on the outside and more on the inside, water will leave the cell and cause it to shrink. That's all osmosis is. It's just the movement of water through the cell membrane. Active transport is using ATP energy to pump things into or out of the cell. And we already talked about the proteins that can do that. And lastly now, we have a little example of osmosis for you. We have 3 beakers here. Inside each beaker is a little yellow cell and embedded in a liquid. And if you look at the first one on the left there, you'll see it says cell with 90% water inside of it, which is mostly what most cells are. And then you have, it's floating in a 80% 20% starch solution. Now the starch can't get through the membrane because if you remember, starch is a big polysaccharide. It's too big to get through there. So it's just going to stay on the outside. But that means there's 80% water outside and 90% water inside, which means that for every 9 molecules of water that leak out of the cell, only 8 leak in. So what's going to happen here is that the cell will lose water and it will shrink. And it'll eventually it might even die from this. If you put a blood cell to this solution, it would look like a raspberry. It would just crinkle right up as it loses its water. The side of the cell membrane that has a higher percentage of water, that's the hypotonic side. The side that has the lower percentage of water is the hypertonic side. So water always goes from where there's more to where there's less, so it goes from hypo to hyper. If you look at the second beaker, you'll see it has 90% water in the cell, but now it's floating around in only water, 100% water. And what's going to happen there is for every 100 molecules that leak in, only 90 leak out. So the cell is going to constantly gain water. Eventually, it will literally explode because of the pressure of the extra water going in. Animal cells can't handle that. They will literally explode. If you put your blood cells in 100% water, you could actually watch them explode under the microscope. Plant cells, however, have a problem because they deal with fresh water all the time. When it rains, rain is almost 100% water. And so what happens, it goes up the plant, goes into the leaves, and bathes those leaf cells with almost 100% water. And so in a plant leaf, and water tends to want to go into the cell faster than it leaks out, that middle beaker there. That's going to cause water pressure in the cell. So that means around each membrane of a cell in the plant, there's another structure around the membrane. That structure is made of cellulose, and that is the cell wall. And the cell wall is actually there to protect the cells from exploding after it rains. And then finally, you have like an cell situation. In our case, we have 90% water cells, but our liquid that's bathing the cells inside of our bodies is also about 90% water. And so that means for every 90 molecules that leave, 90 molecules go in. It doesn't seem to change much. And so what happens, the cell is stable. And that's why we don't need a cell wall, because we're not bathing our cells in almost 100% water like ants do. We are bathing ourselves inside of us with a salty fluid that actually is about 90% water. And so the cell does not change. And so we stay alive without a cell wall. And lastly here, we have one more. When you have a cell like a white blood cell, white blood cells can actually change their shape. And that's what's happening in the first one, is that there's a Golgi vesicle in there with something inside of it. And it's migrating to the outside of the cell, and the cell is opening up and let that material out. That's a great example of secretion and how Golgi works. When something is leaving the cell like that, they call it exocytosis. Exo meaning exit, it's leaving. On the other hand, a white blood cell here on the right, it's taking something in. It could be bacteria that's trying to infect you, and the white blood cells literally eating it. And what will happen is that it'll close up around the bacteria or virus or whatever it is, and it goes inside of a food vacuole or a food vesicle. That will actually merge with a lysosome. The enzymes in the lysosome will actually digest and destroy the bacteria or virus that was trying to hurt you. And that's how white blood cells try to protect you from being sick. Okay, so anyway, that's the story of cells for this week. Check out the quiz for next week. Now you have 1 week to take the quiz. Make sure you do it sometime between now and Friday. It's going to be not online anymore after Friday. So you have an hour and a half to do 30 questions, and you can use your survival guide to do it. It should be very easy for you to do. It's worth 15 points. So make sure you get it done this week as soon as you can. Okay, and when you hit submit, it will be automatically graded and put into your grade book for you. Okay, so you have a great week this week. Do the quiz 1, and also do the questions, survival guide questions, like you do with the assignments, and hand them in too. So you have 2 jobs this week: cell questions and the quiz 1. Okay, you guys have a great week, and we'll see you next time around. If you have any problems, we'll talk about it on Wednesday at 6.